Good day, everybody. My name is Tanya Murky-Epp, Project Manager with the Alliance Coordinating Office. On behalf of the Canadian HIV Vaccine Initiative Research and Development Alliance Coordinating Office, I'd like to welcome you to our webinar today. There's a few of you that haven't been to one of our webinars, and for you who aren't familiar with us here at ACO, we were established by the Government of Canada and the Bill and Melinda Gates Foundation in November 2011 at the International Centre for Infectious Diseases, which is a not-for-profit, non-governmental organization based in Winnipeg. And you can find out more about us and our partners at our website, alliance-aco.ca. So today's webinar, entitled Clinical Implications of HIV Adaptation to Immune Selection, which has been presented by Dr. Sabrina Brume, um, um, who is an associate professor in the Faculty of Health Sciences at Simon Fraser University. This webinar will provide Canadian researchers and members of the Canadian HIV Vaccine Initiative Research and Development Alliance with an overview of HIV mutational adaptation to host cellular immune responses and the implications of this for HIV immune control, HIV prevention, and reservoir elimination. The webinar will be of particular interest to HIV researchers and trainees who are interested in a broad introduction to this topic, as well as an update on recent advances in the field. As always, everyone is welcome. There will be an opportunity to ask questions during the Q&A at the end of the webinar. And during the Q&A, you may let us know that you have a question by raising your hand and unmuting your microphone or using the comment box that will be available on the lower right side of your screen. But before we begin, we have a few questions of the audience to help us to better understand who you are. So I'm going to pull up a few questions. There's 5 of them. So I'm going to start with the first one. Okay, the first one is, do you consider yourself to be a new and early career investigator? And click on yes or no. Okay, about half. Okay, great. And our second question is, how is your audio connected for this webinar? Are you using a telephone or are you using your computer or internet? Okay, terrific. Almost 90% are connected via their internet. Okay, terrific. Our third question is, which research pillar do you most identify with? Basic science, clinical science, epidemiological science, or social science? Okay, mostly basic. Okay, thank you. Our fourth question is, in which sector are you currently working? Academic research, industry research, with an international organization, government or community organization, or other? Okay, great, thank you. And last question, here we go. So if you are viewing this webinar as a group, how many people are viewing this webinar together with you? And you can just type it in there. Okay, we'll give you just a couple more seconds. The screen there for you. Okay, the learning objectives for the webinar today are that you'll gain understanding of how HIV mutational adaptations to host immune responses have been systematically cataloged, a review of recent findings that reveal significant negative consequences of acquired and transmitted HIV adaptation on clinical prognosis, and thirdly, there will be a discussion of the implications of HIV adaptation to antiretroviral-based HIV prevention strategies and HIV cure research. Okay. Okay, so it's my pleasure to introduce to you our speaker, Dr. Sabrina Brume, um, who has received her PhD from the University of British Columbia in 2006 and subsequently completed a postdoctoral fellowship at Harvard's Reagan Institute. She is currently an associate professor in the Faculty of Health Sciences at Simon Fraser University and an associate scientist at the BC Centre for Excellence in HIV/AIDS. Her research integrates molecular biology, epidemiology, and computational approaches to examine questions at the interface of HIV-1 virology, immunology, and pathogenesis, with the ultimate goal of providing new avenues for vaccine design and strategies to eradicate viral reservoirs. So thank you very much for being here, Sabrina. I will now turn it over to you.
Thanks, Tanya, and thanks to everyone for joining this morning. Let me see. How do I advance? There we go. Okay, so this is just the overview of what I'm going to be talking about in the next 40 minutes or so. It's been quite apparent for the last 20 years. HIV definitely is able to mutate to evade detection by cellular immune responses. We've known that for a long time, but until relatively recently, I would argue that the actual impact of immune escape, meaning the clinical implications of this for people and for populations, haven't been completely clear. So what I want to do today is review relatively recent data indicating that HIV immune escape, or I'm going to be calling it HIV adaptation, is actually a bad thing. It's associated with impaired immune responses, elevated plasma viral loads, and faster CD4 T cell decline. And this is true whether you select escape in your own autologous virus or if you are transmitted a virus that's already pre-adapted to your HLA to some extent. And I'll talk a little bit about the implications of this for vaccines. And after that, I'll touch upon some broader implications of HIV adaptation, namely implications for eradication strategies, but also for PrEP, which might come as a surprise to some. So I'll go through the background, which is approximately the next 10 slides or so. I'll go through a little bit quickly, and then I'll spend a little bit longer going through the actual data. Oops, I'm supposed to use these buttons. There we go. Okay. So this is a cartoon showing the process whereby HIV is able to mutate to escape recognition by cellular immune responses. So the panel on the left is so normally if a virus infects a cell, that cell is equipped with an indicator system to alert the immune system that it is infected with a virus. And that indicator system is referred to as the HLA, or human leukocyte antigen molecules. And these molecules are able to travel back and forth between the inside and the outside of the cell. And while they're inside the cell, they'll pick up little bits and pieces of proteins that are being synthesized in that cell and display on the outside. So if the cell is infected with a virus, these HLA molecules will pick up little bits and pieces of viral proteins, display them on the outside of the cell for recognition by cytotoxic or killer CD8 positive T cells. But as shown on the right, what HIV is able to do is to select mutations in its genome that change the sequence of its viral proteins slightly, such that they are no longer presented by these host HLA molecules. And this allows the infected cell to evade detection by cellular immune responses. And what this cartoon on the right is showing here is that the result of that is that viral variants or escape mutant HIV, or I will call today adapted HIV variants, these are released from that infected cell and go on to infect new ones. So HIV is able to mutate to avoid detection. So this is one of the major reasons why our immune system is not able to control or get rid of HIV. But one thing we've come to appreciate in the last, 15 years or so is the specific mutational pathways that the virus takes in order to evade detection by these HLA-restricted immune responses are extremely predictable. Okay, so which means that if we know what HLA class 1 alleles a person is expressing, we can, to a certain extent, predict what mutations are going to happen where in the HIV genome, and to a certain extent, when. And one of the ways in which we've been able to do that, meaning one of the ways in which we've been able to systematically identify what mutations occur in the virus selected by which HLA class 1 alleles, is through large genetic statistical association studies, where we've studied cohorts, large cohorts of HIV-infected people for whom HLA class 1 typing has been done, and for whom viral sequencing has been done. And we've simply identified the mutations in the virus that are significantly overrepresented in people that express certain HLA class 1 alleles. And we've done this over the last decade or so. And what we have now is pretty comprehensive and detailed maps of all the sites in the HIV genome that commonly mutate in response to specific HLA class 1 selection pressures. And that's what's shown here on this slide, is what we call an immune escape map of a protein called HIV-1 GAG, and blown up at the bottom here is a particular epitope that is known, or a particular epitope meaning one protein fragment in the HIV GAG protein that is known to be presented by HLA B57 and B58, two particular HLA alleles. And here we see a very, very common mutations that occur within this epitope at position 3 from T to N and position 9 from G to A. So these are very, very common and reproducible mutations that are selected exactly here in the HIV protein in response to these, in people that have these particular HLA alleles. And we've done this now for HIV subtype B and a bunch of other subtypes. So this slide here is just showing studies in the last 5 years or so that have mapped these HLA-associated adaptations in the virus in different HIV subtypes, in different cohorts, in different places around the world. So this is just to say that we now have a very good idea of how the virus mutates in response to specific selection pressures. So really, where we're at with respect to identifying these immune-mediated mutations, we're pretty much at where the resistance field was 20 years ago. Okay, so starting about 20 years ago, the HIV resistance field began to comprehensively map what resistance mutations are selected by what particular antiretroviral drugs, and there's an international panel, and they update their maps every year. And this information is really important for clinical practice, because these are the lists of mutations that we look for when we're doing personalized genotypic resistance testing to guide treatment decisions for individual HIV-infected persons. But also these are the mutations that we look for when we're doing epidemiological surveillance to see how much transmitted drug resistance might be circulating in a particular population. So one of the things that I just wanted to bring up today is that finally, with immune-mediated changes, we're at this point where we now have lists of them. Okay. However, in contrast to the drug resistance field, so the drug resistance field is ahead of us, because not only have they mapped where all the drug resistance mutations are, they showed pretty early on that resistance was bad news, right? It was bad news clinically, and it's bad news for transmitted drug resistance in the populations. It was really clear really early on that these drug resistance mutations were very clinically important and relevant. The thing is, for these immune-selected polymorphisms, yeah, we know where they are now, but up until recently, it's actually been surprisingly difficult to actually demonstrate that escape mutations are bad for you clinically, okay? And it's been tough to demonstrate that in the case of people who select them during their own infection, and it's been also relatively difficult to show that if one is infected with a virus that's already, to some extent, pre-adapted to one's immune response. We'd think it would be pretty straightforward to be able to demonstrate, but it has been surprisingly hard. So I'll just go through some reasons. So the next couple slides just show why it's how and why it's been kind of difficult. So it's been shockingly difficult to show that after escape mutations are selected in a person, that there's a viremia consequence of that. Again, we'd think it would be straightforward to show, but it's been difficult. There have been some papers that have shown a temporal correlation between escape and then viral load increase, and this is one of them here, published by Philip Golder's group 20 years ago now. But still to this day, studies like this are in the minority. For most epitopes that escape, it's been difficult to actually demonstrate that there's a viral load increase. In fact, we've been far better at documenting why it's complicated to try to show this than we've been at actually trying to show it in the first place. So here's just a couple of papers documenting, you know, the complexity of immune escape. So, for example, over 10 years ago now, Todd Allen's group were among the first to show that after an immune escape mutation is selected in HIV, in many cases, the person's immune response is able to sort of regroup and then target that newly selected variant, right? And that's called de novo CTL responses. And when that happens, it makes it more difficult to detect, you know, a viremia increase. More recently, we've actually shown that de novo CTL epitopes come up. So when there's escape in one epitope, sometimes that actually leads to the creation of another immunogenic epitope right nearby, which is then able to be targeted. So that's another reason why it's been difficult to demonstrate on a per epitope basis. Another big reason it's been difficult is because of fitness costs, right? Some escape mutations come at a cost to viral replicative capacity, so that would offset the magnitude of viral load increase. But suffice it to say, it's been surprisingly difficult to show the relevance of these on an individual epitope level. You would think that, you know, okay, well, maybe it's hard to show it on a per epitope basis, but my goodness, we should be able to look at sequences and count how many escape mutations there are, and then globally show that a higher burden of escape in any given person is associated with poor clinical prognosis. And here I feel confident, you know, sort of poking fun at this particular study by our group nearly 10 years ago. Yes, we can show that a higher proportion of immune escape is probably clinically worse for you, but the magnitudes of these correlations in this study, and, you know, others that I'm not mentioning, are pretty weak, right? They're not particularly convincing. So here we are at this point, while we've done a really great job of mapping where all these polymorphisms are, we've had a hard time, you know, demonstrating their relevance, and, you know, not by way of making excuses. But one thing that's been a challenge with population-based studies is, let's just say we're looking at a specific, you know, an HIV sequence in a particular person that has the relevant HLA. Okay, so in this case, I'm using as an example somebody who has HLA B27, and here we're looking at the sequence of this very well-known immunodominant epitope in GAG. And let's say, if we look at this person's sequence, they have wild-type sequence, meaning there's no escape in this particular epitope. Well, if you're just looking at sequence data and you don't have any other information, well, there could actually be two reasons for this. First, one reason for having wild-type virus at a specific epitope is because that person's mounting an extraordinary immune response against that epitope, and the virus hasn't managed to escape it yet, right? On the other hand, there's a diametrically opposed reason that somebody could have wild-type virus at a particular epitope, and that's because, despite them having the relevant HLA, they don't have T-cell receptors that can target it. Okay, so these are two completely diametrically opposed scenarios with respect to the consequences for viremia, because on the left-hand scale, that would be associated with control, meaning excellent responses, no escape yet. That's associated with control. Whereas if you're incapable of mounting a response against that epitope to begin with, that would be really poor consequences for immune control. And then the complicated thing is that if you have escape, it actually indicates that at some point you were able to target that epitope, but you've escaped in the meantime, so you're somewhere in the middle. So because wild-type virus is, either on one end or the other end of the scale, when we do these population-level studies and all we're looking at is the virus sequence, we lump those two things together and we basically negate our own ability to find associations to begin with. So it's just been complicated. So what we realized as a field is that we needed a few things to come together before we can actually demonstrate conclusively and convincingly that immunoscape is actually bad. And from there, hopefully, we can move on to incorporating this information into vaccine design. So the three of the important things that we've needed is, first, we've needed to map, these mutations across the viral genome. I think we've done that pretty well. The second is we need some kind of better metrics for quantifying adaptation of a given sequence to a given person. Previously, we were just counting escape mutations, and that worked reasonably well. But the thing is, all escape mutations aren't created equal and aren't as common. So we need a new, more nuanced metric that takes into consideration the strength of selection of that mutation in the person with the particular HLA. And we've actually accomplished that now too. And the third thing we need is just more data and more feature-rich data. So we need large data sets, both longitudinal and cross-sectional, from cohorts around the globe for which not only HIV sequencing and HLA genotyping has been done, but clinical data are available and CTL response data are available, at least for some of the cohorts. And I think we've waited until now to be able to answer these questions because we hadn't had all these things all together. So what I want to do for the bulk of this presentation is to go over the results of this paper that was published by a large collaboratory last year, because I think this is one of the first papers to more comprehensively and, in my opinion, convincingly show that immunoscape is actually bad news. So I'll walk you through some of the major highlights of this work. So the first tool that made this particular study possible is the development of a metric to quantify how adapted a specific HIV sequence is to a given person's HLA profile. This metric is more nuanced than previous approaches that simply, counted the number of mutations. This metric is more nuanced in that it weights the specific mutations according to the probability of that mutation would actually be selected in that person with the HLA, basically weighting them according to the strengths of selection at the population level, such that an escape mutation with really, really strong data supporting its association with that HLA would be weighted more strongly than one that is, less common or a weaker association. And this metric scores HIV sequences on a scale from -1 to 1, where -1 is a theoretical minimum and -1 is a theoretical HIV sequence that is absolutely, completely not adapted at all to that person's HLA profile. And the theoretical maximum of 1 is an HIV sequence that is completely adapted to that particular person. We actually never see sequences that score -1 to 1, but that's the scale. So 0 is just in the middle of this scale. 0 doesn't mean no adaptation. 0 just means in the middle of -1 to 1. So the first thing we needed to do was actually to try to show that our metric was actually measuring something, and that ideally that it was measuring the level of adaptation of a given virus to a specific person. So we did this with a number of different cohorts, and here is just some data from a cohort in British Columbia of approximately 2,000 individuals. And here what we did is we quantified the level of adaptation of a person's virus to their own HLA class 1 profile. And the distribution of that is shown in red. We call that autologous adaptation. So on average, the level of adaptation of a person's HIV to them registered at 0.27 on this scale from -1 to 1. Then we quantified something called heterologous adaptation, and that we just randomly selected someone else's virus and quantified what the level of adaptation of that randomly selected virus from someone else was to that patient, and the distribution there is shown in blue. Okay, so there's a nearly there's a difference of approximately 0.5 of this total scale that goes from -1 to 1. Okay, so this is providing evidence that the metric is measuring something, and specifically it's a measure of subject-specific viral adaptation. But one could argue that this is just cross-sectional data. So what we actually really need to do is show this longitudinally, and we did this too, and we did that using a cohort of Zambian transmission pairs. And here the rationale is when a person, a recipient, acquires HIV, at the moment of transmission, at that moment, that virus will actually be more adapted to the virus, to the HLA alleles of the donor, right? Because the donor is the one who just transmitted the virus to the recipient. So at the beginning, the virus should look like the donor's HLA, but over time, that virus should develop mutations that make it more and more and more adapted to the recipient. That's what we hypothesized. And that hypothesis was borne out in an analysis of linked transmission pairs from Zambia, where here we see adaptation to the donor HLA declining over time, and adaptation to the recipient's HLA class 1 alleles increasing over time. Still, we acknowledge that at 2 years post-infection, the virus still looks a little bit more like the donor than the recipient, but we imagine by the 3 or 4 year mark, the lines will cross. So these data were just to set the stage for arguing that our metric of adaptation was actually measuring something, was measuring adaptation in a meaningful way. The first thing we set out to show is that if you have HIV and you develop escape mutations, that's bad for clinical prognosis. And we did this in a number of ways. The first way is through analyses of just correlative analyses. So here we had access to a number of cohorts from the Americas. We had a cohort of elite controllers from North America, and 2 cohorts of chronically infected individuals, so non-controllers, one from the United States and one from Canada. And what we did here is we quantified the level of adaptation of the viruses in these cohorts to the patients in these cohorts. And regardless of regardless of where the cohorts were, we saw that the level of adaptation of HIV to the host was way, way, way lower in elite controllers compared to, chronically infected individuals. And this was true when we looked at everyone, but it was also true when we only specifically looked at people exhibiting the protective class 1 alleles B57 or B27. So the overall results are not dominated or driven by the enrichment of protective alleles among elite controllers versus non-controllers. Rather, the metric of adaptation is significantly correlated with measures of control, where people who control HIV naturally have very, very, very low levels of adaptation of their virus to their HLA class 1 alleles. I don't have a slide for this, but we did numerous other analyses. And the most important is that in an analysis of nearly 3,000 chronically infected individuals, the level of adaptation to the virus, to the person, that was the single most important predictor of viral load and CD4 T cell counts, even after controlling for all the normal sociodemographic, viral, and host factors that one would normally control for. So adaptation, meaning low adaptation to the host, is a correlate of being able to control viremia. The other thing that we found, again, just looking at chronically infected cohorts, is that once you escape essentially any protective effects that are associated with having, protective alleles, they are lost. Okay, so this is a slide here showing HLA-specific effects on plasma viral load. In this case, it's a cohort from Southern Africa. And usually when you see these graphs, we normally see, expression of a particular HLA allele on the y-axis, and then, viral load with respect to the population median on the x-axis. So it's the same kind of graph, except we've further stratified the people that have each HLA allele by how adapted their virus is to them. Okay, so again, the usual suspects, protective alleles, are at the top, and then the non-protective ones are at the bottom. But what I want to draw your attention to is the fact that the protective alleles are only protective if the virus hasn't adapted to them yet. So for example, if I use, say, B5703, which is the second one from the top as an example. The average viral, if you have B5703 and your virus has not adapted to B5703 yet, you have a viral load reduction of nearly 1 log compared to the median. But if you're B5703 and your virus has adapted to B5703, there's essentially no residual viral load benefit anymore. Your viral load in that case is the same, indistinguishable from the population mean. And this is true for a number of the well-known protective alleles. Okay, so HLA alleles are only protective until the virus figures out how to escape from them, and then they're not protective anymore. This also, I'll come back to this later, but there's also been a debate a really long time. It's like, oh, well, maybe some of the protective alleles they're protective because they select for escape mutations that cause, big hits to viral fitness. And, maybe that is associated with some residual viremia benefit. But this analysis here would suggest that that's, not really the case, but I'll come back to this. I keep pressing the wrong button. And here's another analysis, also in this case incorporating analyses of viral adaptation, but also incorporating information from immune responses. Okay, so here again is cohorts of chronically infected individuals, and we're still trying to tease apart, is escape really bad for you? And now we're bringing in information about your immune response. So this on the x-axis here, we divide the cohort based on whether their sequences have low adaptation to the patient's HLA profile or high, very high adaptation, so lots and lots of escape to the patient's HLA profile. And then we further stratify within those categories by the strength of the immune response, okay, as measured by interferon gamma ELISpot in vitro. Red is patients who have not very good CTL response breadth, but blue are folks that are mounting really great and broad CTL responses by ELISpot. One of the most important things to take away from this figure is that the bar that is the lowest, so the group that is the most associated with viral control, are the people who have great immune responses, broad immune responses, and for whom the virus hasn't completely escaped yet. That is the second set of dots, the second set of blue dots, right? This is significantly lower than all the others. Even if you have, broad immune responses, but your virus has, managed to escape most of those immune responses, that's the furthest bar to the right. The viral load is not any different from the population median. This graph also argues for a lack of important fitness effects. If you actually compare the two red groups of dots, they are not significantly different from one another, right? If you would suppose that a very, very, very high burden of escape mutations would be associated with, perhaps some viral load residual benefit. The second red bar should be lower than the first one, but in this case, it's not significantly different. But nevertheless, the major take-home point of this slide is that the best viral load control is if you have great CTL responses and your virus hasn't managed to escape yet. That is true in chronic HIV infection. So moving on, hopefully I was able to convince you that escape, if it's selected in a given patient, is bad news clinically. But the other thing that we're wondering about is transmission of escaped variants. Okay, how does that affect clinical outcome? So that was the next part of this study. So again, here we went back to the Zambian transmission pairs cohort, and we quantified the level of adaptation of the presumed transmitted founder virus to the recipient. Okay, and we stratified the cohort into people who acquired HIV that wasn't really all that adapted to their HLA profile yet. That's the blue line. And people who acquired HIV that was quite highly adapted to their HLA profile already, and that's the red line. And this is a simple Kaplan-Meier analysis that shows that acquiring a virus that's already pretty adapted to your HLA profile is associated with significantly faster CD4 decline, okay, or the lack of the ability to maintain CD4 counts above 250. The same is true for negative consequences with respect to viral load. So here again, we're looking at the Zambian transmission pairs, and we're looking at the level of adaptation of the virus when it's transmitted to the recipient. The more adapted a virus is to the recipient, the higher the set point viral load. Okay, the Spearman R isn't ultra strong, but nevertheless, this is a significant. It's a significant association. And again, I'm not showing you the data, but adaptation of the presumed transmitted viral founder sequence was the best predictor, or predicted the most amount of the population variation in CD4 decline and set point viral load, predicted way better than any other normal things that we would look when we were looking for predictors of clinical outcomes in people recently acquiring HIV. This is a little bit of a tangent here, so I'll try to explain this slide. There have been a number of reports in the last decade or so that have made the observation that plasma viral load looks to be, to a certain extent, heritable. And what is meant by that is that phylogenetically, people who acquire similar looking viruses tend to have similar viral loads, almost as if there was some inherent viral genetic property that's contained within the virus sequence that influences what the viral load is going to be. So, for example, maybe it's some kind of fitness, intrinsic replicative capacity, right? There's some aspect of the virus that is that. So the associations are not, they're usually not strong, but they're certainly there. But this has been controversial, in part because different studies have quantified the extent of this heritability. That was not clear. There are a wide range of estimates of the extent of heritability. That's what I mean. So some studies say, yeah, there's some heritability, but it's around about 2%. So why do we care? Whereas other studies have said, no, heritability, it's up to about 60%. So it's been very study specific and cohort specific. But if we now apply sort of an adaptation lens to these studies, we can now start to tease this apart. So what I'm showing here are three graphs. Again, we're looking at the Zambian transmission pairs. and I apologize for the typo. The header should say low adaptation similarity, medium adaptation similarity, and high adaptation similarity. So what we've done here is we've quantified the extent to which the donor and recipients HLA class 1 allele profiles select for similar pathways of adaptation. Okay, so meaning if the donor and recipient have similar, the same or similar class 1 alleles, the transmitted virus is going to be highly adapted to both the donor and the recipient, right? So what we've done here is we've actually stratified the entire cohort by the similarity of the donor and recipient's HLA alleles, and thus the level of the extent to which the virus is going to be adapted to both of them. If there's a big mismatch, right, meaning that the donor and recipient have HLA alleles where the adaptation is very different, meaning the donor and recipient, where there's a incompatibility, a virus that's adapted to the donor is not going to be adapted to the recipient. That's on the left. That's the low adaptation similarity. But once you get to donor and recipient pairs who have very similar HLA profiles and thus would share a lot of the same ways of virus adaptation, then we see that there is an excellent correlation with viral load heritability. So this was a little bit of a tangent, but we think in this case that adaptation can help explain a lot of these dichotomous findings, and it's actually not necessarily an inherent property of the virus, but rather a measure of the virus's level of adaptation to certain HLA alleles in a given human population. So I'll come back to this. I think this is my last slide. No, it's okay. Sorry. Yeah. Never mind. With respect to transmitted adaptation, we were also able to show this not only in the Zambian transmission pairs, but also through reanalysis of data from the STEP trial. We were able to show that the level of adaptation of any given virus to a given recipient was associated again with higher viral load set point. So why is it when we acquire a virus that's already adapted to our immune responses, right? Why is acquisition of adapted viruses, associated with poor clinical prognosis? Well, this probably might not come as a surprise, but it's because CTL responses against these pre-adapted, viral variants are not as good as those against wild-type virus, and in some cases, they actually look like they're not only not good, but actually dysfunctional. And we showed this in a number of ways. One way is we had 11 acute early infected individuals for whom we were able to reconstruct the likely founder virus. And then we scanned these founder virus sequences for optimally described epitopes within them. And then once we found those optimally described epitopes, we synthesized them and classified them as to whether they were non-adapted to the patient. Those are shown in blue, or adapted to the patient, and those are shown in red. And we measured by interferon gamma ELISpot, and broadly there was a significant result in that people's CTL responses were way better at recognizing non-adapted compared to adapted forms of the epitope. Okay, so perhaps not surprisingly, adapted epitopes are not as immunogenic as non-adapted ones. And in fact, if we do this on a per patient level, and that's what's shown on the left here, if we quantify the extent to which, the epitopes in the autologous founder virus are already adapted to the patient at transmission, there's a significant inverse correlation, right? The more adapted a person's virus is to them, the worse the CTL response rate will be to the epitopes in the incoming virus. And we usually show these data to people, and people think, well, of course, they're adapted, right? A lot of these mutations abrogate the ability of these peptides to bind to the HLA to begin with. So this is not a surprising result, is it? And perhaps not, but one thing that we wanted to point out is that we found a number of cases actually where the adapted version of the epitope was still predicted to bind to the HLA very well. In fact, in some case, better. So we thought, oh, well, maybe the epitope predictions are crappy. So we actually experimentally verified the ability of these adapted epitopes to bind to the HLA, and in some cases, they did bind better than, or the same or better than the other one. So it's not necessarily a problem with binding and presentation to the HLA. In some of these cases, we were also able to isolate CTL clones in populations that were specific for either the non-adapted or the adapted form. And in each of these cases, we always found that the ability of CTL specific for the non-adapted form of the epitope to kill HIV-infected cells was better than the ability of CTL that recognized the adapted form to kill infected targets. And this is an experiment here just with a representative epitope. And in this case, the targets were cells infected with the HIV MJ4 infectious molecular clone for clade C that just differed by one amino acid in the entire proteome. And it was the adapted version in this particular epitope.
Sabrina, it's Tanya. I just wanted to give you a quick time check. We've got about 5 to 6 minutes left before we need to open it up to questions.
Oh, okay. In this case, well, maybe I'll see if I can get to the first summary slide, and then maybe we can just stop there. Why don't I try that?
Okay, sounds good.
Would that be okay?
Yeah, I think that'll be fine. If people can stay longer, just type it in the chat box. Otherwise, we'll go ahead with Sabrina's plan here.
Yeah. Actually, this is pretty okay. Well, okay. I'll try to. So, anyways, I'll keep going and I'll stop when it's time for questions. The next thing we wanted to be able to show is that HIV adaptation is actually, well, it's likely to be eroding the ability of HLA to control HIV in some global regions. And this is because it's become apparent in the last few years that, escape mutations are selected and then they get transmitted from host to host, and they don't all necessarily revert, right? If these adaptations persist, even in people who don't have the relevant HLA, we might actually have a problem where, as the pandemic progresses, we're getting accumulation of adaptations in HIV in circulation, meaning that as the pandemic progresses, HIV is adapting more and more and more to us. And if that's the case, this is bad news immunologically. And to a certain extent, well, we think that's happening faster in some populations with respect to others. So here are data from Southern Africa and Canada, and here is at the population level. And what's being shown here is on the x-axis. So each of these dots on each of these graphs is a particular HLA class 1 allele. They're not labeled, but each of them is this particular allele. And we're plotting the average level of adaptation of the average circulating virus sequence to that HLA alongside its protective capacity, so the average reduction in plasma viral load that allele usually gives you. And regardless of where we look in the world, we see a significant trend where the more adapted the average virus sequence is to a particular HLA, the less protective that HLA allele is. So in other words, protective HLA alleles are those to which the circulating viruses haven't adapted yet. And this is extremely region specific. So in this particular analysis, we have 4 alleles, particular HLA alleles that are, sometimes associated with being protective, especially in subtype C. But here we're looking at their average protective ability in 3 different cities in sub-Saharan Africa: Durban, Lusaka, and Gaborone. And then so the dots are color-coded by what allele they are. So just look at B51. B5801 is protective in Durban. but it is not protective in Gaborone or Lusaka. In fact, it's only protective in Durban because in Durban, the average circulating virus isn't highly adapted to B5801. But in Lusaka and Gaborone, the average circulating virus is very highly adapted to B5801, and B5801 is not protective anymore in those two cities. So this is the ability of an HLA to be protective is highly specific to a population. There's now other studies starting to come out that are corroborating these results. I just wanted to touch upon this particular study from Japan. Japan is an interesting population because there's relatively restricted HLA diversity, and there's certain HLA alleles that are highly overrepresented in the population, and actually escape mutations to those HLA alleles are consensus in Japan. And one of those is A24, and there's this Y to F mutation that happens in the NEF gene. And in this particular study, researchers inferred the transmitted founder virus in A24 expressing individuals, and showed that A24 individuals that had been infected with adapted virus at this residue maintained lower CD4 counts over the entire course of study follow-up than those who had been infected with the wild type version. So numerous studies are coming from different parts of the world that are starting to show this now. And here is probably where I'll just stop here as the summary part one and vaccine implications. This is most of the talk anyways. So here I'll just summarize to say that we think that we've now more convincingly shown that escape is bad for you. So acquiring a founder virus that's pre-adapted to one's HLA profile that undermines initial CTL responses and accelerates disease progression. And the worst news is in some parts of the world, the average level of adaptation of the average circulating virus is, slowly getting higher and higher as the pandemic progresses. So I just want to end this part on talking a little bit about implications for vaccines. So we don't have an HIV vaccine yet, a prophylactic HIV vaccine, and one hypothesized reason why we don't is because the strategies that we've used so far don't adequately account for HIV diversity and immune escape. So different strategies to deal with immune escape and diversity have been discussed for quite a while now. And one strategy is to, build diversity into the vaccine to begin with, to try to elicit, immune responses to diverse viral variants. And I think these new data cast, some doubt on that as a strategy. I'm not saying we should discount that strategy, but our data show that adapted variants aren't as immunogenic as the non-adapted ones. Also, in some cases, and our data don't explicitly show this, but it's possible that they might actually be detrimental or taking away from the immune system's ability to recognize wild-type variants. So instead, these data might more support approaches that target restricted sets of highly conserved epitopes or regions. So conserved element strategies, with the caveat, of course, that there are no completely conserved elements in HIV, and even the leading conserved element vaccines are also, polyvalent vaccines that do have some amount of diversity built into the immunogen. The most immediate, I think, implications for vaccine design, however, is that now we have a metric for adaptation, and we've shown that it's really important. And we strongly suggest that these metrics for adaptation are incorporated into vaccine analyses, so analyses of observation on clinical studies moving forward, because adaptation is one of the most important predictors of clinical outcome. So we should be checking for it because we think that'll help us make sense of data from trials moving forward. In addition, we should be doing things like assessing the adaptation of specific vaccine inserts to the people who become infected. And one of the things that I didn't have time to talk about today is we did such analysis in the STEP trial. We reanalyzed the data from the STEP trial and showed that there was a trend. It wasn't significant, but the extent that the viral insert, the vaccine insert, was already adapted to the patient's HLA, that was associated with a trend towards poorer immunogenicity in those patients. So I'll leave it there because this is the summary of the first part, and the second part is just extra stuff. So hopefully I didn't go too much overtime.
No, you're fine, Sabrina. I posed the question to on the chat, and there seems to be interest in you continuing with the second part. And we'll forward the evaluation link. And if people need to leave, then they're welcome to. But if you have time, and those who are able to want to stay tuned, we can continue on since we've got everybody connected now. If that's okay with you, if you have the time to continue with the second part.
Yeah, I do. Thanks, and sorry for I was talking to him. I'll try to go. I'll just the last part. So I wanted to shift a little bit and be a bit more broad. And so I want to talk very briefly about the relevance of adaptation to HIV cure strategies, and also for PrEP. But I'll do this faster, and I have less data. This is more I'm hand waving now. But recently, there's a lot of attention in HIV cure, and recently it's become apparent that we're probably going to have to worry about immune escape in the reservoir because, well, it's there. And this study by the Silicano group analyzed reservoir sequences from people on long-term HAART and showed that in every case where they isolated a reservoir sequence, there was at least one epitope that had a major escape mutation in it. So they concluded that we were going to need a broad CTL response to be able to eliminate the viral reservoir. But we've been thinking about this too, and that if we now understand that seeding of the reservoir begins, early in infection and continues thereafter, right? So theoretically, the reservoir should actually recapitulate the history of evolution in a given patient, where the reservoir is seeded really, really, really early following infection, should have a lower burden of escape than ones seeded later, meaning that the reservoir should actually be heterogeneous with respect to, what escape mutations are there. And in fact, we should see certain epitopes that are escaped in some, but not in others, depending on when they were seeded, with respect to the individual's history of evolution. So we're just starting to look at this now, and I just wanted to share some data from one patient. So here we have a patient whom we've been studying. This person, the dates look really weird. But anyways, this patient had a lengthy history of pre-ART history from 1996 to 2004. And after that, went on ART, and pretty much has been suppressed for the last 10 years. And so all the icons, the little diamonds, show time points where we were able to sequence HIV from that individual to look at viral evolution. And then we also sampled the reservoir, recently after so many years on ART. And really what we're trying to get at here is, how does HIV evolve in a person? And then what does the reservoir look like, right? Does the reservoir recapitulate what happened in the years previously? And do we see examples of unescaped and escaped versions of the same epitope? And if so, which ones predominate? So here is just a highlighter plot of what's going on in NEF over the time period sampled in this patient. These are all SGA, single genome sequences. And the point that I want to try to make is that, well, in some cases, there we go. Here, for example, is a site that is likely to be a site of adaptation, right? This site escaped somewhere in the middle of 1996, and after that, there was a selective sweep, and almost all sequences after that had that particular mutation. In the reservoir, that's the same, except for you see one reservoir sequence at the bottom that still maintains the wild type. So if this were an escape mutation in this putative B13 epitope, the reservoir would be 96% adapted. Well, that's bad news for trying to eliminate that epitope, trying to eliminate the reservoir with that epitope. But if you look at other epitopes, maybe it's not so bad. So look at this one. The reservoir is 92% adapted, presuming that this is an important escape mutation. But here are some other ones. The reservoir is about 50% adapted. So the point that I just want to make with this is that adaptation probably is relevant for eradication strategies, but what we have to do is gain a much better understanding of the distribution and burden of escape in the reservoir. We actually, just because of an epitope is escaped in some reservoirs doesn't necessarily mean that all of them. So agreed that a broad CTL response is likely going to be needed for this, but just to raise the issue that adaptation is also important and that the metric can be applied to this scenario as well. And then the last thing that I wanted to talk about was PrEP. With this, so at the beginning of the talk, I talked about antiretroviral resistance mutations and how those have been mapped and how we now are understanding immune, adaptations. And in the vast majority of cases, drug resistance mutations and immune escape mutations do not overlap, meaning there are very, very few immune resistance mutations or adaptations that also serve as resistance mutations. However, there are some notable exceptions, and I think we need to pay attention to these exceptions. One of them is a mutation in reverse transcriptase that is selected by HLA-B18, okay, where there's an escape from E to a bunch of other things at this particular site. It's selected by B18. But that also, those specific B18 escape mutations also confer high-level resistance to a newish NNRTI called rilpivirine. So you might be thinking, okay, well, sure, but there's lots of other NNRTIs, so why should we care? But the reason that we should care is because currently rilpivirine is one of 2 antiretrovirals under consideration for PrEP because it can be formulated into long-acting injectable formulations, right? Rilpivirine and cabotegravir, which is an integrase inhibitor, they're currently in clinical trials for, once-a-month injections instead of oral therapy. And for that reason, it's a particularly attractive candidate for PrEP. But this potential fact that there's naturally occurring immune escape variants that would render you resistant to rilpivirine, this might be a problem. I don't want to spend too much time on this, but recently I've been working with a group in Japan, and we've been able to show that, well, B18 frequency differs substantially in populations around the world. And actually, if you look at the prevalence of this reverse transcriptase 138X immune escape variant, there's a highly significant positive correlation between the frequency of that mutation that's selected by B18 and the frequency of B18 in particular populations, where in some places in this world, for example, some countries in sub-Saharan Africa, Botswana is shown on this slide, but Zimbabwe is another, the frequency of E138X is more than 10%. I mean, that theoretically means that if you used rilpivirine as a single agent PrEP, right, in these places, it would only protect, 10% of people wouldn't be protected, right? Because 10% of circulating viruses have rilpivirine resistance mutations that are actually naturally selected by B18 in the population. And to show this a different way, in that particular study, we tried to make this point a different way. Here is a world map. We pulled data out from various public databases as well as cohorts that we had access to, and we colored this map based on the prevalence of E1